Welcome to Ask Prep, your trusted companion for NEET and JEE preparation. Today we will study about a very important topic of biology, which is molecular basis of inheritance, DNA structure, replication, transcription. Let's begin with the structure of DNA, the molecule responsible for storing and transmitting genetic information. DNA, or deoxyribonucleic acid, is a long, thread-like molecule that carries the genetic instructions used in the growth, development, functioning, and reproduction of all living organisms. It is composed of two long chains of nucleotides twisted into a double helix shape. This iconic structure was discovered in 1953 by James Watson and Francis Crick, with the help of Rosalind Franklin's X-ray diffraction images. The double helix structure is often compared to a spiral staircase, where the steps are the pairs of nitrogenous bases, and the handrails are the sugar-phosphate backbones. Each nucleotide, the basic building block of DNA, consists of three components: a sugar molecule, a phosphate group, and a nitrogenous base. There are four types of nitrogenous bases in DNA: adenine, A, thymine, T, cytosine, C, and guanine, G. These bases form base pairs in a very specific way. Adenine pairs with thymine and cytosine pairs with guanine. This pairing is held together by hydrogen bonds. Adenine and thymine form two hydrogen bonds, while cytosine and guanine form three, making the CG pair stronger than the AT pair. Now that we know the structure of DNA, let's move on to one of the most crucial processes in genetics, DNA replication. DNA replication is the process by which a cell makes an exact copy of its DNA before cell division, ensuring that each daughter cell gets an identical copy of the genetic material. This process is vital for growth, development, and the maintenance of living organisms. The process of DNA replication begins at specific locations on the DNA molecule, called origins of replication. The double helix is unwound by an enzyme called helicase, which breaks the hydrogen bonds between the base pairs, separating the two strands. This creates two single-stranded DNA templates that will be used to synthesize new complementary strands. Next, an enzyme called primase synthesizes short RNA primers on each template strand. These primers are necessary because the enzyme that synthesizes DNA, known as DNA polymerase, can only add nucleotides to an existing strand. Once the primers are in place, DNA polymerase begins adding complementary nucleotides to the template strands. On the leading strand, DNA polymerase moves in the same direction as the unwinding of the helix, adding nucleotides continuously. However, on the lagging strand, DNA polymerase works in the opposite direction, creating short fragments of DNA known as Okazaki fragments. These fragments are later joined together by another enzyme called ligase. In this way, both strands of the DNA are replicated, producing two identical molecules of DNA. The accuracy of this process is critical, as even a single error can lead to mutations that may cause diseases or disorders. Fortunately, DNA polymerase has a proofreading function that helps to correct most of the errors during replication. Having understood DNA replication, let's now focus on transcription, the first step in the process of gene expression. Transcription is the process by which the genetic information stored in DNA is copied into a messenger RNA, mRNA, molecule. This mRNA then carries the genetic code from the DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are synthesized. Transcription is crucial for the functioning of cells, as proteins are essential for nearly every cellular process. Transcription begins when an enzyme called RNA polymerase binds to a specific region of the DNA known as the promoter. The promoter acts as a signal to start transcription. Once RNA polymerase is bound to the promoter, it unwinds the DNA and begins synthesizing the mRNA strand in the 5-3 direction, using the DNA template strand. As RNA polymerase moves along the DNA, it adds ribonucleotides A, U, C, and G, where uracil, or U, replaces thymine, that are complementary to the DNA template. For example, if the DNA sequence has an adenine, A, the RNA polymerase will add a uracil, U, to the mRNA. As the RNA polymerase moves along the DNA, the newly synthesized mRNA strand elongates. Once the RNA polymerase reaches a specific sequence of bases known as the terminator, it stops transcription and releases the newly formed mRNA molecule. This mRNA is a copy of the gene that will be translated into a protein. After transcription, the mRNA undergoes several modifications before it can leave the nucleus. These include the addition of a 5' cap and a poly-A tail, which protect the mRNA and help it bind to ribosomes for translation. Additionally, the mRNA undergoes splicing, where non-coding regions called introns are removed and the remaining coding regions, called exons, are joined together. The mature mRNA then exits the nucleus and travels to the cytoplasm for protein synthesis. The process of transcription is highly regulated. Different genes are transcribed at different times and in response to various signals, ensuring that cells produce the right proteins at the right time. This regulation is essential for cell function and the overall development of an organism. In summary, the molecular basis of inheritance involves 3 key processes: the structure of DNA, the replication of DNA, and the transcription of DNA into RNA. DNA's double helix structure allows it to carry genetic information in a stable yet adaptable form. During DNA replication, the cell makes an exact copy of its DNA, ensuring that genetic information is passed on accurately during cell division. Transcription then converts the information in DNA into mRNA, which will be used to synthesize proteins. Thank you so much for being a part of the Ask Prep community. We truly appreciate your time and dedication to learning, especially when it comes to mastering biology, a subject that's not only essential for exams like NEET and JEE. If you found this video helpful, please give it a thumbs up, share it with your friends who are also preparing, and make sure to subscribe to our channel so you never miss an update.